Good morning. I'd like to thank Dr. Michelle Callows, American Society of Gene and Cell Therapy, and the chairpersons for a kind invitation for me to come speak here at this distinguished society meeting. My name is Puri. I'm the director of the Division of Cellular and Gene Therapies in the Center for Biologics Evaluation Research in the Office of Tissue and Advanced Therapies. In my talk, I'll cover the approval of some of the cell and gene therapy products. I'll discuss some of the challenges and opportunities in the manufacturing of cell and gene therapy products. We'll talk about the 21st Century Cures Act and the new designation was created through this act, the Regenerative Medicine Advanced Therapy designation. And I'll talk about the opportunities of interaction with the FDA and our resources and some of the contact information. So the division I come from is the division of cellular and gene therapy. The first chart on the left is the division which deals with the chemistry, manufacturing, and controls information of all cell and gene therapy tissue engineering products submitted to the FDA. We evaluate a plethora of products that include stem cells, stem cell-derived products, somatic cell therapies, therapeutic vaccines, and other antigen-specific active immunotherapies that include cancer vaccines and immunotherapies and personalized vaccines. In gene therapies, we have a variety of different products that include genetically modified cells, such as CAR T cells, vectors, oncolytic viruses, xenotransplantation products, and combination product that includes tissue engineered and regenerative medicine products, devices with a cellular component, and devices that are used to process or select cell and tissue or to deliver cell or gene. The other division in our office deals with the recombinant protein, purified recombinant proteins for hematology indications. I've shown here this chart to tell you the submissions of INDs and IDEs over the years, and I'll focus your attention to last three bars on the right side to make a point that from 2016 to 2017, number of submissions have increased from 223 to 316, 42%. And from 2017 to 2018, increased by 32%, with total of 87% increase from 2016. In the cell therapy submission, that also increased from in 2016 from 101 to 129, which is about 27.7%. And from 2017 to 2018, that increased from 129 to 154 by 19.4%, thus a 52% increase from 2016 to 2018. Gene therapy INDs, the first recorded gene therapy IND was submitted to FDA in 1990. This is about the time I came from Dr. Steve Rosenberg's lab and joined CBER at that time. So the first gene therapy IND was submitted in 1990 for the treatment of for the metastatic melanoma. Overall, the three gene therapy INDs were submitted that particular year. I would like to focus your attention to last three bars. The gene therapy submissions increased 34.2% from 2016 to 2018, but they changed, whoppingly increased for 94% from 2017 to 2018, from 106 to 206 submissions. In 2017, we approved the first gene therapy file, cells, CAR T cell therapy, Kymriah, for the treatment of certain children and young adults with B-cell acute lymphocytic leukemia. In the same year, we approved another drug called Yescarta, with the second product approved for the treatment of adult patients with relapsed or refractory large B-cell lymphoma. In the same year, we licensed a first-in-class adeno-associated virus vector, Luxturna, for expressing the gene for human RP65. You just heard from Dr. Kathy Hai from Spark Therapeutics, as indicated for the treatment of patients with confirmed biallelic RP65 mutation-associated retinal dystrophy. We also approved a variety of cell therapy products. Provenge, Sequelucel T, a autologous antigen-presenting cell vaccine, cancer vaccine. We approved HPC, hematopoietic progenitor cell cord blood, from 8 different cord blood centers in the United States. We approved MACI, which is autologous cultured chondrocytes on porcine collagen membrane for repair of single or multiple symptomatic full-thickness cartilage defects of the knee. And we licensed Gentuit, is a allogeneic cultured keratinocytes and fibroblasts in bovine collagen for surgically created vascular wound bed in the treatment of mucogingival condition in adults. Since the approval of cell and gene therapy products, there has been an explosion of interest in gene therapy, but one of the rate-limiting factors to development has been availability of vectors. In fact, there are a number of articles shown in this slide describing the bottleneck created by the need for gene therapy vectors in academic research. Since the advances in gene therapy to date have been incubated in academic centers, this is indeed an issue and perhaps an opportunity for this community. Beyond the challenges that folks have in finding a facility in manufacturing their vector, we have observed difficult trade-offs between short-term and long-term development of products. In order to get the phase 1 studies off the ground, the choice of manufacturing facilities is sometimes based on cost and availability rather than whether the manufacturing process can actually be scaled up for later phase studies or for commercialization. This has led to the situations where products that look very promising in early stages get delayed because the manufacturing process were never designed to be scalable. We view the product development as integral part of the clinical development. Although this is a more general slide, a stepwise approach that can apply to any product, it is a particular challenge for an academic investigator to ensure that CMC development keep pace with the clinical development, and the scale-up of the production is feasible for later clinical studies or for marketing. CMC development can be challenged even with the traditional clinical development strategy shown here, but we see some gene therapy sponsors wanting to use early phase 1 or phase 2 data to support the licensor, especially in rare diseases where large clinical trials are not feasible. In such cases, it is important to develop the manufacturing process as early as possible, and this includes early development and qualification of test methods. Sometimes major manufacturing changes the product so much that we can be certain that it is the same product. And unfortunately, this may lead to need for more clinical trials. Now I will talk about some of the challenges and opportunities in the manufacture of gene therapy vectors. We are hearing that there are long backlogs of manufacturing lentiviral vectors and adeno-associated viral vector in particular. There has recently been a flood of investment in manufacturing capacity, which is a good thing, but it takes time to build new facilities, to hire and train the appropriate staff. As I mentioned before, that there is still a significant amount of manufacturing being done with non-optimized manufacturing process that ultimately prove to be difficult to scale up. But we believe that there is a significant opportunity for those who design their vector manufacturing process from the ground up to be efficient and scalable. Examples of building efficiency into the process from the start include, as shown in the lower part of the slide, include selecting cells that produce the highest amount of vector and growing them in the modern bioreactors instead of tissue culture flask. This allows increased cell density and reduces the need for manual processing. Downstream purification can also be optimized to reduce loss of vector, for example, during chromatography or during sterile filtration. This is a challenging message, given that there is an immediate need for manufacturing. But ultimately, we believe that field will best be served by designing efficient, scalable manufacturing processes that can be used from phase 1 all the way to the higher phases of clinical studies and commercialization. Cell and gene therapy products are complicated products, unlike small molecules or small chemical drugs. And this is especially true for autologous cell therapies that are manufactured in small batches from highly variable patient cells. This manufacturing has to be done on a very tight schedule because of the short half-life. To expand the capacity, manufacturing of these autologous therapies can only be scaled out because it cannot be scaled up. This means that manufacturing facilities need to have many individual manufacturing areas and lots of manufacturing staff. Current cell manufacturing processes tend to require significant manual input, and this could be a significant benefit from increased automation. Another interesting area, or with great potential, would be to develop allogeneic cell therapy products instead. These therapies are sometimes called universal or off-the-shelf of the product, and it should be possible to scale up manufacturing to fit the size of the patient populations. Researchers are developing ways to get around some of the challenges with allogeneic cell products, especially reducing the rejection by the immune system by using cutting-edge technologies such as genome editing. To facilitate the manufacturing and development of cell and gene therapy products, we plan to invest in CBER's research program and in collaboration with our stakeholders with the goal of improving the efficiency of cell and gene therapy manufacturing. Increasing manufacturing efficiency can help to address shortages by allowing greater yields of product within the same manufacturing footprint. We also think that there is a significant potential for technologies and manufacturing methods that reduce the number of manual processing steps. This can only lower the cost of manufacturing and increase product consistency at the same time. I'm showing here some of the examples of possible improvements, such as cell lines that have better productivity or can be grown to higher density, vector production methods that don't cause as much loss of vector during purification, and advanced automated cell culture technologies that reduce the need for manual processing, and a development of advanced assays for characterization using high throughput technology for identification of critical quality attributes. In CBER's research program, we are currently searching for new lead researchers in the area of advanced manufacturing, as well as building on existing expertise within our program for improving cell and gene therapy products. FDA has published a guidance document that describes the availability of various expedited programs as shown on this slide for product development, including breakthrough therapy and designation. These programs offer enhanced interaction with FDA that facilitates product development for serious conditions. United States Congress passed an act, 21st Century Cures Act, that was passed by the bipartisan support of both houses of Congress by 392 to 26 votes in the Congress and 94 to 5 votes in the US Senate. And it was signed into the law by the President Obama on December 13, 2016. Through this act, a new designation pathway was created called RMAT, Region Medicine Advanced Therapy Designation. Region Medicine Therapy products included, as described in section 3033, include cell therapies, therapeutic tissue engineering products, human cell and tissue products, and combination products thereof. FDA interpreted this law by way of guidance document as provided here that includes cell therapies, gene therapies, including genetically modified cells that lead to sustained effects on cells and tissues, and xenogenic cell products. So Region Medicine Advanced Therapy Designation is a drug is eligible for this designation if it meets the criteria I just mentioned in the products I mentioned in the previous slide, and the drug is intended to treat, modify, reverse, or cure a serious or life-threatening disease or condition. And the preliminary clinical evidence indicates that drug has the potential to address unmet medical needs for such condition, for disease or condition. The benefits of RMAT designations are summarized here. It provides an early interaction with FDA to discuss any potential surrogate or intermediate endpoint to support accelerated approval. Similar to breakthrough therapy designation products are also given quite a bit of interaction for their manufacturing of issues during their development. These RMAT designated products may be eligible for priority review. They may be eligible for accelerated approval as agreed upon during product development, which is based on perhaps surrogate or intermediate endpoint, which are reasonably likely to predict long-term clinical benefit. Any sponsor can make a request with a new IND submission or amendment to an existing IND. FDA has 60 calendar days to determine if designation criteria are met. We provide the written response If the RMAT designation is not granted, we'll provide you in writing the description of the rationale why the RMAT designation was declined. The request for the designation should include how the drug meets the definition of region medicine therapy, how the drug meets the criteria it is intended to treat, modify, reverse, or cure a serious or life-threatening disease or condition, and that preliminary clinical evidence indicates the drug has potential to address unmet medical need for such disease or condition. So as of March 24, 2019, we have granted 32 products RMAT designation. 54 were denied and 7 were still pending. Most of the RMAT designations were given to products, neurology and hematology and oncology indications, followed by cardiovascular and others. The total number of submission of RMAT designation requests were, majority of them were cell therapy products, which were allogeneic cells, donor-derived cells, followed by gene therapy products and cell therapy products with autologous cells, followed by combination products where cells, the device or cells and genes and combination thereof. So I have listed here some of the slides why, some of the reasons why some of the breakthrough RMAT designation requests were declined. There were some administrative reasons that the IND was not in effect, it was already inactivated, and there was no preliminary clinical evidence submitted. There could be CMC reasons that the proof of the preliminary clinical studies were performed using a product, and after those study, that product had undergone so many manufacturing changes, and it was hard to determine the product was same, or it had changed, and the sponsor has not provided compatibility data to tell us that it is the same product. Or there will be study design issues or inconsistent results and the product activity, that some effects may be positive while the other effects were negative, where actually the product was hurting the patient. In those cases, it was difficult to determine the clinical evidence of effectiveness. And I'd like to share with you that FDA has, CBER, has started a new program called Interact for early communication with CBER. This is an initial targeted engagement for regulatory advice on CBER products, previously called as pre-IND interactions. The goal here is to obtain early feedback on product development program and say for a novel and innovative investigational agent. It is a mechanism for early communication with OTAT. We give non-binding recommendations. It's an informal scientific discussion between the reviewers of all discipline from the FDA and the sponsor. It is initial targeted discussion of specific issue. For example, if the sponsor has done some proof of concept studies and some safety studies, and they haven't done a pre-pivotal preclinical studies, they would like to get some advice on their large study. We do not generate any meeting minutes. And what is out of the scope of this Interact program is questions about candidate product selection for further development, or situations in which the sponsor previously has received formal regulatory advice about a similar product and indication. So I listed here that last year, in 2018 July, we published 6 gene therapy guidance documents. The first one deals with the chemistry, manufacturing, and control information for human gene therapy, INDs. The second one is the RCR testing of retroviral vector-mediated gene therapy products. And the third one deals with the long-term follow-up after administration of gene therapy products. And we published 3 disease-targeted guidance documents for hemophilia, retinal disorders, and rare diseases. We received large number of comments from our stakeholders, and we are incorporating those comments in this guidance to finalize them, to publish them soon. I've listed here 2 very useful sites for you to visit. And the first one deals with the regulatory processes in our Office of Tissues and Advanced Therapies. And the second one is the OTAT Learn webinar series. This is a wonderful series of little videos given by our subject matter experts that includes in regulatory project management, chemistry, manufacturing, and controls, the pharmacology, toxicology studies, the clinical trial design issues, and some of the regulatory issues and regulatory questions. So I would recommend it highly, so you can view them in the luxury of your offices or in your homes. So together, and in summary, we are committed to bringing the promise of innovative, safe, and effective new therapies to those who need them as quickly as possible. I would like to, lastly, but not the least, I'd like to acknowledge the colleagues in the Division of Cellular and Gene Therapies with whom I have a pleasure to work with for their incredible dedication and hard work to keep up with the ever-increasing number of submissions to our office. I'd like to recognize our office leadership team, Drs. Brian and Anatol, and our colleagues, Dr. Wang and Zhu, in helping me provide some of the slides. And I'd like to put a little plug here. By the way, we are now recruiting. So we have received some positions through 21st Century Cures Act, through Prescription Drug User Fee Act. So it is a pleasure to recruit folks, and we have a booth downstairs in the exhibit hall. I encourage you to come out and talk to our folks. We'll have a number of our experts available to speak with you for the wonderful opportunities that we have in our office. And we're recruiting all disciplines of folks. And I'll leave you here with my contact information. And also, if you have any regulatory questions, you want to send an email, there's a link here, Consumer Affairs Branch, Manufacturing Assistance and Technical Training Branch, and there you can write the question, and they get the input from our SMEs, subject matter experts, and give you the answer as soon as possible. And you can always write to me. My email number is provided here. And of course, I'd like to thank you for your kind attention, and perhaps with the permission of chairs, we'll answer any questions you might have.